Hello, and this is going to be a review of Unit 6, which we talked about the cell cycle and mitosis. And you can see it's broken down into two parts, although we may have actually broken it down into three. This is a post-unit review. I think we actually put cancer and the causes and prevention actually in either B2 or it was in C, I'm not exactly sure. But you can see what we're going to do is we're going to talk about analyzing the cell in terms of size and volume and why it's limited, compare its ability to transport materials based on the ratio of surface and volume. Then we're going to talk about the diagram of the stages of mitosis, which we called PMAT, and then you're going to describe interphase, mitosis, and cytokineses, which I called PMAT-C, and we're going to compare the cycles of plants and animals, a couple of differences between the two, one because they've got a cell wall and we don't have to worry about it, and we have centrioles, and they do the same thing without centrioles, and prokaryotic cells and eukaryotic cells, and then we're going to define cancer in terms of prevention and things. Okay, so the first slide looks like this, and what we did is we talked about different boxes, and I had the kids actually draw the dimensions and figure out this is a 1 by 1 by 1. So we have 1 cubic millimeter of volume, and then we have a 1, 2, 3, 4, 5, and 6 in terms of surface area. So we actually divide surface area by volume and came up with a 6. Did the same thing with a 2 by 2 by 2. Come up with 8. We had 24. Now we've got a 3 surface to volume, and the number's getting smaller and smaller. And then we had a 4 by 4 by 4. And what we basically found out is that as the cell got bigger, the volume, so the surface area volume ratio got smaller, and since we used the outside of the cell to get things in and out, we wouldn't be able to get things in fast enough, and we're also getting farther away from the nucleus, so the nucleus can't get supply out. Then I showed them the difference between, and then, well, we did this one where there's 6. We do 5 by 5 by 5, and we actually come up with a 1.2. And you can see this one goes from a 6 to 50. That would be a 25 times larger, and this one's 125 times larger in terms of volume. So you can see the volume changes much faster than the surface area, and that's what the cell has to do. If you take that and you actually break it up into the same size boxes here, you still have the surface volume ratio of 6. Then we talked about the cell cycle, or actually the phases of mitosis. We started with interphase, the normal cell, where we have two centrioles sitting next to each other because one had just been copied from the other. We have DNA is actually the strands of DNA, which is called chromatin, and they're doubled. So if this was a human, we have 23 pair or 46 chromosomes in there, each being about 6 feet long. You can do the math, there's a lot of DNA. It's all in a nuclear envelope or nuclear membrane, which has pores in it, so we're going to talk about that in Unit 7, where we actually get RNA moving out in terms of mRNA to actually do protein synthesis. Then we move into mitosis, first phase called prophase, and remember P, MAT, C. And what we do is we see the centrioles moving out, parts of the cytoskeleton called the spindle fibers are starting to form. The chromatin actually comes into condensed form, which are called chromatids, and you can see there are two sisters, they make up the X. Then we actually get those moving apart. We get the nuclear envelope starting to break down. You can see just very little of it here when we get to late prometaphase, which is late prophase, early metaphase. The chromosomes start lining up in the center. Spindle fibers reach into the centromeres, which is the thing that's actually holding the two things together. They're calling them something different, but we learned them as centromeres. And then when we get to metaphase, where they are lined up, the two sister chromatids are held together by centromeres. Spindle fibers have grabbed onto the centromere. Centrioles have moved to the poles of the cell. Then we actually move to anaphase, where the centromere gets dissolved. The two sister chromatids get pulled apart. They're now called daughter chromatids. They're still called sisters, though. One's moving up to the top part, this centriole, the other one's moving down to the bottom pole, which is this centriole, and then we have the cell membrane actually pinching off for cytokinesis, which ends in mitosis and starts the next phase, interphase. Plant cells a little differently because of the plant wall. You can see a normal plant cell. We're missing the centrioles we had here for animal cells, but we have no centrioles. We have the DNA as a chromatid, as the strands. They actually come in as the sister chromatids, the X-shaped. Spindle fibers form, but they're not forming from centrioles. The nuclear envelope breaks down. They line up for metaphase. They get pulled apart in anaphase. And then when we get to, telophase, chromosomes start to dissolve again back into chromatid. We get a cell plate starting at the center. Can't pinch in because it's a cell wall. And here you can see it in a real plant cell. They're lining up, pulling apart, and then there's that cell plate where instead of cytokinesis, we still get cytokinesis, but we don't get the pinching in, the cleavage furrow. We get a cell plate. Then we talked about the cell cycle. Here's the PMAT. There's cytokinesis, where we're breaking up the cytoplasm as well as the organelles so that we actually have two new cells. This cell ought to be half the size of this cell because when G1 we actually start growing. And you can see in G1, which comes right after cyto, we're moving in a clockwise position right after cytokinesis. This is where we actually double the cell contents. So we get twice as much cell membrane, twice as much cell cellular plasm or cytosol. We get twice as many Golgi bodies, endoplasmic reticulum, ribosomes. We get twice as many of everything. Some cells come out as what's called GO. Heart cells do this once, nerve cells do this a few times. Other cells come out and they will not go through the rest of this phase because they're done. They're actually doing what they're supposed to do. If there's a problem with this, the cell will pull it out and lyse it, but most of the time when we get to GO, this is where the cell arrests, the cell cycle arrests, which basically means this cell is not going to go through mitosis again. Heart cells are a real good example of that. And then we move into S phase, which is where we actually we synthesize the DNA, we double the DNA so we can actually have chromatids, because at this point we actually have a chromatid in each one. We don't have the sisters, so we can break them apart in mitosis. So we double the DNA. For humans, the 23 chromosomes that are in each one gets doubled to 46 chromosomes for the 23 pair we need to actually go through mitosis. And then we get to G2, and this double checks everything. Did mitosis work out? Did it grow enough? Did we get enough of the chromosomes? Did we get the chromosomes done correctly? And it'll try to repair everything it can. If it can't, it's going to kick it back out and lyse it. But if it actually is okay, it moves into mitosis and starts the process all over again. Interphase, which you can see is this blue part of the circle. 80 to 90% of the time is spent on interphase, where the cell's actually doing its basic cellular job. If it's a bone cell, a muscle cell, a skin cell, a connective tissue cell, a lung cell, it's doing its job here in this process for interphase, as well as going through all these G1, S, and G2, where we double the contents, double the chromosomes, and double check everything. Then we have C, PMAT-C, which kicks the whole process of mitosis, splits into two cells so we can start it all over again. Here's a cell cycle animation with 5 questions, and I'm going to go ahead and click it, and hopefully it'll open up here kind of quickly, because I know this is getting kind of long. We only have one more slide to go after this. This is on my teacher computer at school, and they've told me that they're going to give me new RAM. As you can see, this is taking way too long, and I don't think I can stop it. I can't. I'm sorry for this. And we're just sitting here spinning. Isn't this cool? So it opens up the Internet Explorer, which is not the best bet. Okay, this is coming from, you can see McGrath-Hill.com. It's actually their student view, how the cell cycle works. It's supposed to push play. It's got some text here. I'm just going to go through. Now I just lost control of my icon. There we go again. I'm just going to slide this through, I hope. As you can see, nothing is changing. Oh, there we go. Let's try this a little slower. Okay, we go through, we talk about the whole cell cycle. We break a cell into two, we go through interphase, centrioles move apart, and then we move through the phases. Centrioles move apart, spindle fibers form, chromosomes line up, they get ripped apart, centromeres dissolve, they get ripped apart at anaphase, they move over, we get the cleavage furrow, we get 2 new cells, identical cells, called daughters, half the size, and then we move into G1 synthesis, S, and then G2. And then there are some quizzes down here, like which one represents the correct order of the cell cycle, G1, S, G2, and M. Division of cytoplasm, cytokinesis. Which of the following represents mitosis, PMAT, and then DNA replication. In mitosis, that's false because it's in the S phase and interphase. Mitosis and genetically cells, that's got to be true, and you can submit it, and you can see that you got 5 correct. Excellent. Okay, last slide we have to do is this one. Excuse me. Okay, what happens when mitosis goes wrong? If mitosis goes in and we actually get the wrong type of cell coming out, cancer cells, they lack the stoplight, so they don't know when to stop. They just keep going through this and going through this and going through this. And mitosis, we can have it working perfectly, and then we can have a mutation in one cell where these 3 keep going, producing heart cells or lung cells or skin cells. This one's going to actually be a mutation, and it's going to be a mutation that actually causes cancer. There's the cell we're talking about, and it actually starts to grow. You can see it's a different shape. They grow much faster than the regular cells. They actually start producing parts of the cell. If it's malignant, that is going to grow in and invade other parts of the cell by getting into the blood vessels and moving. But we also, in order to get a cancerous tumor to work, we have to get something called angiogenesis, where these cells are going to want more and more material, oxygen, carbohydrates, lipids, things like that. So what it does is it puts an angiogenic factor down, the blood vessel grows, and blood goes in to feed all those new cells. Okay, thank you much for stopping by. Appreciate it. Longer than I wanted it to be. Bye.